Good morning, students. This is Murthy sir. Till now, we discussed about bacteria, algae, fungi, protozoans, microarthropods, viruses. Another interesting microorganisms are there. They are called blue-green algae or cyanobacteria. They are the first formed living organisms in oceans millions of years back. So students, today we will start with blue-green algae. They are also called cyanobacteria. Students, just like bacteria, blue-green algae are also prokaryotic organisms. They contain chlorophyll pigment, they can photosynthesize. Blue-green algae, they undergo asexual reproduction, just like budding we have seen in East. And this blue-green algae, they form blooms in ponds and lakes. This blue-green algae can survive in extreme conditions like hot springs. And another interesting feature is this blue-green algae can perform nitrogen fixation, just like rhizobium and other bacteria. It can convert atmospheric nitrogen gas into nitrogen compounds. Let us see some examples of blue-green algae: nostoc, Glyocapsa, Chlorococcus spirulina. So students, some forms of blue-green algae, they survive in the soil and make the soil fertile. Here you can see some examples of blue-green algae: nostoc, Glyocapsa, Chlorococcus spirulina. All these are different forms of blue-green algae. Now let us see in detail what is nitrogen fixation. Students, in atmosphere, 78% nitrogen is there. It is a diatomic molecule. Nitrogen we write as N2. And this nitrogen gas was discovered by Daniel Rutherford. This nitrogen gas will not react easily with other chemicals. That's why we call somewhat inert in nature. So plants, they take the help of bacteria or blue-green algae for the conversion of atmospheric nitrogen gas into nitrogen compounds like ammonia and nitrogen salts like nitrates and nitrites, so that the root system will be able to absorb them along with water. Students, now let us move on to the world of medicine. Antibiotics are mainly used to treat bacterial infections. Earlier in the previous class I told, first antibiotic was synthesized by Alexander Fleming. It is penicillin. He extracted it from a fungus named Penicillium notatum. Antibiotics, they kill the bacteria or inhibit the growth of bacteria. Now, in modern medicine, many kinds of antibiotics are available. Antibiotics are mostly extracted from bacteria or fungi, and some are synthetic ones prepared from chemicals by pharmacists. Antibiotics should be used after consulting a doctor. So as per doctor's prescription, we have to use them, and excess use of antibiotics show some side effects on our body, so we have to be very careful. Students, he is Dr. Yellappragada Subbarao. He was a great Andhra scientist who did research in USA, and he synthesized some important antibiotics, and he contributed for chemotherapy, cancer treatment. He is called the Wizard of Wonder Drugs. Dr. Y. Subbarao synthesized tetracycline, which is used for many bacterial infections. Students, tetracycline, earlier it was used for the treatment of plague infection, which is caused by infected rats. Dr. Y. Subbarao spent most of his life in USA conducting research in medicine and finally passed away there. And now let us see the importance of vaccines. What is a vaccine? A vaccine is prepared basically from killed or weakened germs of that particular pathogen which is causing the infection. It is prepared in the form of a suspension. Vaccines basically prevent the cause of infection. They are injected into the body of a healthy person before getting the infection so that his body will be ready with antibodies to fight against the germs which are responsible for a particular infection. Students, great microbiologist Louis Pasteur synthesized vaccine for rabies infection, which is a viral infection. This rabies infection is caused by the bite of mammals, mostly infected stray dogs. Students, a person who is bitten by an infected stray dog or a mammal should take treatment immediately. Otherwise, this rabies virus attacks the nervous system and it may lead to death. Hence, pasteurization is named after great microbiologist Louis Pasteur, who developed this preservation technique. Mostly it is used for preservation of milk and some food items. This pasteurization is used in dairy industry for preservation of huge amounts of milk, which will be delivered to people every day. Pasteurization technique is used in dairy industry to preserve the milk, to make it free from germs which spoil the milk. In this process, they heat the milk to 71 degrees centigrade for just 15 to 30 seconds. Students, in ultra high temperature pasteurization, they heat the milk to 140 degrees centigrade for just 2 seconds. So it is called UHT process. Dr. Verghese Kurian is called the milkman of India, who contributed for the development of dairy industry in our country. Our Indian government sent Dr. Verghese Kurian to USA for higher studies and research. Now let us learn how food poisoning is caused. It is caused by a bacterium called Clostridium botulinum. This food poisoning results in abdominal pain and vomitings, nausea. When food poisoning is caused, immediately the patient should consult the doctor and he should take proper treatment for that. Students, to avoid food poisoning, we must consume always properly cooked food items, and when we consume packed food items, we have to see the expiry date. That is very important. Now, let us learn about different food preservation techniques. Here you can see drying, smoking, cooling, freezing, salting, pickling, and canning. All these are different types of food preservation techniques. Students, in any food item, if little moisture is there, fungi will spoil that food item. So, when we purchase different food grains, first we have to keep in sunlight. We have to dry them up. After that, the containers which you are using, they should be in dry condition. Then the food will not be spoiled by fungi. Friends, in our homes, when we make pickles, whether it is mango, lemon, tomato, whatever it is, more salt and oil are used as preservatives. Students, you all know about vinegar. It is used as preservative in food items. You know, this vinegar contains a weak organic acid called acetic acid. So this acetic acid acts as a preservative. Students, now we will learn about some common plant and animal diseases. This is called pathology. The scientific study of diseases is called pathology. Some common plant diseases are citrus canker in citrus fruits, red rot of sugarcane, ticca disease in groundnut. These are caused by different bacteria, fungi, and viruses. So agricultural scientists, they advise the farmers to use particular chemicals to prevent these infections. Now let us see some common diseases in cattle. cows, buffaloes. These are reared by villagers, farmers, in addition to agriculture. Anthrax and foot and mouth disease are common in cattle. Bird flu is common in poultry farms. Students, to cure these bacterial, viral or fungal infections, veterinary doctors they give proper treatment to the cattle. Now let us learn about soil fertility. A fertile soil is required for the healthy growth of crop plants. A fertile soil should contain humus. It should contain soil living organisms like earthworms, bacteria, fungi, blue-green algae. It should contain required amounts of macronutrients NPK, nitrogen, phosphorus, potassium, micronutrients. The pH of the soil should be neutral. The soil should contain aeration and water holding capacity. Then only the crop plants will grow well and we will get a good yield. Students, in villages, farmers they will maintain compost pits. A compost pit is a small pit. In that, they will drop all the organic matter, plant and animal waste. It will be dropped in that, and after some days, because of decomposition, it will turn into a rich manure.